Hello everyone, good day. This is Ma'am Honey and today we will be talking about cellular respiration. What is cellular respiration? Cellular respiration is a process present in cells of living things in order to get energy by breaking glucose molecules. In breaking these glucose molecules and other organic compounds, energy stored in their covalent bonds will be released, which will be used in order to produce ATP. ATP is a short-term energy storage which will be used by the cells to do their work. How does cellular respiration occur? Cellular respiration occurs when one molecule of glucose or C6H12O6 reacts with 6 molecules of oxygen, producing 6 molecules of carbon dioxide and 6 molecules of water, releasing energy in a form of adenosine triphosphate or ATP. Cellular respiration involves 3 stages. The 3 stages are glycolysis, Krebs cycle, and electron transport chain. First stage of cellular respiration is glycolysis, where it breaks or splits glucose to form 2 molecules of pyruvic acid, and in the process, ATP and high-energy electrons carried by NADH are produced. The 2 molecules of pyruvic acid will then be converted into acetyl coenzyme A, and in the process or reaction, carbon dioxide will be released. Once pyruvic acid is converted into acetyl coenzyme A, it will then enter to Krebs cycle, and in the Krebs cycle, it produces ATP, carbon dioxide, and NADH and FADH. And these NADH and FADH are electron carriers which are used by the electron transport chain to form or to produce more ATP. What is ATP? ATP stands for adenosine triphosphate, and again, ATP is a short-term energy storage which will be used by cells to do their activities. ATP is an organic molecule containing high-energy phosphate bonds. ATP is composed of the following: a nitrogenous base, a ribose sugar, and 3 phosphate groups. How do we get energy from ATP? We get energy from ATP by breaking the high-energy bonds between the last 2 phosphates in ATP. Once ATP is hydrolyzed, 1 phosphate group will be removed, releasing energy. And this energy will be utilized by cells to do their work. In cellular respiration, NADH and FADH2 are also produced. These 2 molecules are also known as high-energy electron carriers, where NADH is produced from the reduction of NAD-positive, while FADH2 is produced from the reduction of FAD-positive. Where does cellular respiration take place? Cellular respiration takes place in 2 parts of the cell. Glycolysis occurs in the cytoplasm, while the Krebs cycle and electron transport chain take place in the mitochondria. Glycolysis specifically occurs in the cytosol of cytoplasm. Krebs cycle occurs in the mitochondrion matrix, while the electron transport chain occurs in the inner membrane of the mitochondrion. If we are going to review the structure of the mitochondrion, it is composed of a smooth outer membrane, a folded inner membrane, the folds called cristae, and the space inside the cristae called the mitochondrial matrix. Again, glycolysis occurs in the cytosol of the cytoplasm. Krebs cycle occurs in the mitochondrial matrix, while the electron transport chain occurs in the inner membrane or across the cristae. The first stage of cellular respiration is glycolysis. Glycolysis means splitting of sugar or C6H12O6. It occurs in the cytosol of the cytoplasm. In glycolysis, it partially oxidizes glucose, which is composed of 6 carbon, into 2 pyruvates or 2-pyruvic acid, which is composed of 3 carbons. This process occurs whether or not oxygen is present. Glycolysis is an exergenic process, meaning energy is released. Most of the energy harnessed is conserved in the high energy electrons of NADH and in the phosphate bonds of ATP. Basically, in the glycolysis process, glucose is converted into 2 pyruvates of pyruvic acid, and in the reaction, 2 molecules of ATP are formed from substrate level phosphorylation, where a phosphate group is attached to 1 molecule of ADP. And in the reaction, 2 molecules of NAD positive are reduced to form 2 molecules of NADH and 2 hydrogen ions. Glycolysis has 2 phases. First phase is the energy investment phase, and the second phase is the energy payoff phase. In the energy investment phase, ATP will be hydrolyzed, producing or releasing 1 phosphate group and ADP. 1 phosphate group will bind with glucose, producing a product which is the glucose 6-phosphate, wherein 1 phosphate group is attached to glucose. The glucose 6-phosphate will then be converted to fructose 6-phosphate. From fructose 6-phosphate, ATP will then be hydrolyzed. Again, another ATP will be hydrolyzed, producing a phosphate group and ADP. The phosphate group coming from this ATP will then bind to the fructose 6-phosphate, producing fructose 1,6-diphosphate. So meaning the fructose here has now 2 phosphate groups attached. From the fructose 1,6-diphosphate, it will then be converted into 2 molecules of glyceraldehyde 3-phosphate or G3P. So this compound is composed of 3 carbons, and 2 molecules of G3P will be produced in the energy investment phase. So basically, in the energy investment phase, 2 ATPs are hydrolyzed or invested in order to attach phosphate groups in the glucose molecules or sugar molecules. The product of energy investment phase is the 2 molecules of glyceraldehyde 3-phosphate or G3P. And these 2 molecules will be converted into 2 molecules of pyruvic acid or pyruvate. The 2 molecules of G3P will be converted to form 2 molecules of 1,3-diphosphoglyceric acid, in which in the process, reduction occurs where 2 molecules of NAD+ is reduced to form 2 molecules of NADH and 2 hydrogen ions. As you can see, the 1,3-diphosphoglyceric acid is composed of 2 phosphate groups. And these 2 molecules of 1,3-diphosphoglyceric acid will be converted into 2 molecules of 3-phosphoglyceric acid. As you can see, this molecule contains 1 phosphate group. It is because in the reaction, the substrate level phosphorylation occurs, where it is when an enzyme transfers a phosphate group from a substrate to ADP. So basically, 1 phosphate group from 1,3-diphosphoglyceric acid is detached for it to bind with 1 molecule of ADP to produce or form ATP. Since there are two molecules of 1,3-diphosphoglyceric acid involved, that's why two molecules of ATP are formed. From 3-phosphoglyceric acid, or the two molecules of phosphoglyceric acid, it will then be converted into two molecules of 2-phosphoglyceric acid. From the two molecules of phosphoglyceric acid, it will be converted into two molecules of phosphophenol pyruvic acid. And in the process, two molecules of water are produced. From two molecules of phosphophenol pyruvic acid, it will then be converted into two molecules of pyruvic acid, where in the reaction, substrate level phosphorylation occurs. Again, this process occurs when an enzyme transfers a phosphate group from a substrate to ADP, forming ATP molecule. And again, since there are two molecules of phosphophenol pyruvic acid involved, that's why two molecules of ATP are produced. So final product of glycolysis are the two molecules of pyruvic acid, wherein it is equal to one molecule of glucose. To sum up the processes in glycolysis, again, glycolysis has two phases, the energy investment phase and the energy payoff phase, where in the energy investment phase, two molecules of ATPs are invested in order to form two molecules of G3P or glyceraldehyde 3-phosphate. From the two molecules of G3P, it will then be converted in order to form the final products, which is the two molecules of pyruvic acid, where in the reactions, two molecules of NADH, two hydrogen ions, and four molecules of ATP are formed. To sum it up, glycolysis takes place in the cytoplasm, where it is anaerobic, meaning it doesn't use oxygen. Again, in glycolysis, it has two phases, the energy investment phase and the energy payoff phase. In the energy investment phase, it requires the input of two adenosine triphosphate or ATPs. In the glycolysis, one glucose molecule is converted into two molecules of pyruvate, where in the process, two molecules of NADH and four molecules of ATP are also produced. Next to glycolysis is the formation of acetyl coenzyme A. This formation is the junction between glycolysis and Krebs cycle. Again, the two pyruvic acids are oxidized to form acetyl coenzyme A for it to enter Krebs cycle. Pyruvate molecules are translocated from the cytosol into the mitochondrion by a carrier protein in the mitochondrial membrane. In order for these pyruvate molecules to be transferred from the cytosol of the cytoplasm to the mitochondrial matrix by a carrier protein, a carbon dioxide is removed from pyruvate, making a 2-carbon compound. And this 2-carbon compound will attach to coenzyme A, forming acetyl coenzyme A. If we are going to look at the formation of acetyl coenzyme A, one molecule of pyruvic acid forms one molecule of acetyl coenzyme A. In the reaction, one molecule of carbon dioxide is released, producing a 2-carbon compound acetyl. And the 2-carbon compound acetyl will attach with the carrier protein coenzyme A to form the final product, which is the acetyl coenzyme A. Again, it is composed of a carrier protein coenzyme A and an acetyl group. In the reaction, reduction occurs where one molecule of NAD positive is reduced to form NADH and one hydrogen ion. Since there are two pyruvic acid from one glucose molecule, there will be two acetyl coenzyme A that will enter the Krebs cycle. Second stage of cellular respiration is Krebs cycle. Krebs cycle will only occur when oxygen is present since it is aerobic, meaning it requires oxygen. Two molecules of pyruvic acid from one glucose molecule will be converted to acetyl coenzyme A for Krebs cycle to begin or takes place. It is a cyclical series of redox reactions that give off carbon dioxide and produce 1 ATP per cycle. Since there are two acetyl coenzyme A molecules from two pyruvic acid molecules, Krebs cycle turns twice per one glucose molecule. Therefore, in one glucose molecule, two ATPs are formed from Krebs cycle. If you are going to look at the process, one molecule of acetyl coenzyme A requires one turn of Krebs cycle. In the process, it produces two molecules of carbon dioxide, three molecules of NADH, one molecule of ATP, and one molecule of FADH2. Since two molecules of acetyl coenzyme A enter the Krebs cycle will take two turns per one glucose molecule. To know more about the process of Krebs cycle, we have here the diagram. First is the acetyl coenzyme A will get into the Krebs cycle wherein it is composed of a carrier protein attached to a 2-carbon compound acetyl. The carrier protein coenzyme A will release the 2-carbon compound acetyl to react with the 4-carbon compound oxaloacetate to form 6-carbon compound citric acid. The 6-carbon compound citric acid will then be converted into a 5-carbon alpha ketoglutaric acid, wherein it is composed of 5 carbons since in the process, one molecule of carbon dioxide will leave the cycle and NAD positive will be reduced to form NADH and one hydrogen ion. From a 5-carbon compound alpha ketoglutaric acid, it is converted into 4-carbon compound succinic acid. As you can see, one carbon is removed since in the reaction, one molecule of carbon dioxide leaves the cycle. NAD positive is reduced to form NADH and to release one hydrogen ion, and also a substrate level phosphorylation occurs where an enzyme in the reaction transfer a phosphate group from a substrate to one molecule of ADP in order to form ATP. From succinic acid, it is converted into malic acid where in the reaction FAD positive is reduced to form FADH2. From malic acid, it is then converted into oxaloacetate in order for the reaction to start again, where in the process, in the reaction, NAD positive is reduced to form a molecule of NADH and one hydrogen ion. To sum it up, one turn of Krebs cycle. For step 1, acetyl coenzyme A strokes the furnace. In steps 2 and 3, NADH, ATP, and carbon dioxide are generated during redox reactions. And in steps 4 and 5, redox reactions generate FADH2 and NADH. Each turn of Krebs cycle produces 3 molecules of NADH, 1 FADH2, and 2 molecules of carbon dioxide. Therefore, for each glucose molecule, wherein after glycolysis 2 pyruvic acid are formed, the Krebs cycle produces 6 NADH, 2 FADH2, 4 carbon dioxide, and 2 ATPs. Again, in the process of cellular respiration, it has three stages. First stage is glycolysis, followed by Krebs cycle, and then the electron transport chain and chemiosmosis. In the glycolysis process, one glucose molecule is broken down to form two pyruvic acids, and in the process, it produces an ATP and high energy electrons which are carried by NADH. In order for the pyruvic acid to be utilized by Krebs cycle, it needs to be converted into acetyl coenzyme A, and in the conversion, carbon dioxide is released. In Krebs cycle, the acetyl coenzyme A are processed, and in the reaction, ATP, carbon dioxide, NADH, and FADH are also produced, wherein these NADH and FADH molecules are where energy, or these are energy carriers that carry high energy electrons, and this energy in these two types of molecules will be used in the electron transport chain in order to produce more ATP. The third stage of the cellular respiration is electron transport chain. And again, electron transport chain occurs in the inner membrane of the mitochondrion. And this process is aerobic. So there's a need of oxygen which pulls the electrons from NADH and FADH down the electron transport chain to lower energy state. Oxygen here is the electron acceptor. And in the process, it produces 34 ATP, or 90% of the ATP in the body. Electron transport chain is basically the transportation of electrons from one molecule to another, and the final electron acceptor is the oxygen. FADH2 and NADH2 molecules carry high energy electrons, and for every FADH2 molecule, 2 ATPs are produced, while for every NADH molecule, 3 ATPs are produced. The process of ATP production is what we call chemiosmosis. It uses the energy of hydrogen ion gradients across the membrane to phosphorylate ADP. The main component of ATP synthesis is ATP synthase. ATP synthase is an enzyme at the same time a carrier protein which is found in the inner membrane of the mitochondrion. The ATP synthase allows the movement of hydrogen ions from high concentration area to low concentration area. It uses energy of the ion gradient to power ATP synthesis. For every hydrogen ion that flows through ATP synthase, one ATP can be formed from ADP. The electron transport chain is composed of protein complexes wherein electron carriers are found. Again, main goal is to transport electrons from one molecule to another and to transport it until it will reach the final electron acceptor, which is the oxygen. First thing that happens in the electron transport chain is the oxidation of NADH. Again, one molecule of NADH produces three molecules of ATP. In the oxidation of NADH, electrons are produced since there's losing of electrons. Again, these electrons carry high energy, and these electrons will be transported by the electron carriers found in the protein complexes. As electrons are transported from one molecule to another, the energy that it carries will be used up in order to pump hydrogen ions from the mitochondrial matrix into the intermembrane space. High energy carried by electrons will be used to pump hydrogen ions from the mitochondrial matrix into the intermembrane space. With that, there's this what we called high concentration of hydrogen ions. And when there's high concentration of hydrogen ions, passive diffusion occurs. And in passive diffusion, there's a need for hydrogen ions to pass through a protein channel. And the protein channel is what we called the ATP synthase. Again, in every one hydrogen ion, one ATP molecule is produced, wherein the energy will be used up from the gradient will be used up by the ATP synthase to bind one phosphate group to one molecule of ADP, producing an ATP. Again, since we are using the electrons coming from NADH here, final product of this process will be 3 molecules of ATP. The three stages of cellular respiration are glycolysis, Krebs cycle, and electron transport chain, wherein in the glycolysis, glucose molecule is broken down to form 2-pyruvic acid, forming 2 ATPs and 2 molecules of NADH. The 2-pyruvic acid will be used by Krebs cycle after the conversion of these two molecules into 2-acetyl coenzyme A. And in the conversion, it will produce 2 molecules of NADH. In the Krebs cycle's process, 2 molecules of ATP are formed and 6 molecules of NADH and 2 molecules of FADH will be produced, which are used by the electron transport chain to produce about 34 ATP. Again, in one NADH, it produces 3 ATP, while in one FADH, it produces 2 ATPs. So, 2 plus 2 plus 6 is 10. Times 3, it's 30. Plus 2 times 2, it's 4. So, 30 plus 4, so we have 34 ATPs. Plus the 2 ATPs from the substrate level phosphorylation is equal to 36. So 36 plus another 2 ATPs from glycolysis, which is about 38 ATP total. So again, maximum per one glucose molecule, there will be about 38 ATPs. To sum up the cellular respiration, glycolysis produces 2 ATPs and 2 molecules of NADH, which could produce 4 to 6 ATPs, and it depends on how these NADH molecules gets to the electron transport chain. To make things simple, we will say that these 2 NADH makes 4 ATPs. Formation of acetyl coenzyme A, it produces 2 NADH, since in one NADH there's 3 ATPs produced. So 2 times 3, it's 6 ATPs. Plus the 2 ATPs from Krebs cycle, the 6 NADH, which is about 18 ATPs, and 2 FADH, which is equal to 4 ATPs. So total, it's more or less 36 ATPs in one glucose molecule. Another type of respiration is anaerobic respiration. Anaerobic respiration occurs when oxygen is not present. The process is what we call fermentation. There are two types. It could be a lactic acid fermentation, which usually occurs in muscle cells, and alcoholic fermentation, which occurs in yeasts, which produces alcohol or ethanol. There are two ATPs produced in the process or the fermentation process. In the process of fermentation, one glucose molecule will be converted to form 2 molecules of pyruvic acid. In the reaction, 2 molecules of ATP will be formed from substrate level phosphorylation, and 2 molecules of NAD+ are reduced to form 2 molecules of NADH. Final product of glycolysis is 2 molecules of pyruvic acid. And these two molecules of pyruvic acid will then be converted into two molecules of ethanol. And in the reaction, two molecules of carbon dioxide are released and two molecules of NADH coming from the glycolysis will be oxidized to form two molecules of NAD positive. And the final product is ethanol or two molecules of ethanol. Same with the lactic acid fermentation, glycolysis occurs and the final products for glycolysis are the two molecules of pyruvic acid. And these two molecules of pyruvic acid will be converted to form two molecules of lactic acid. And in the reaction, two molecules of NADH will be oxidized to form two molecules of NAD positive. Again, fermentation occurs in anaerobic condition where oxygen is not present and the two molecules of pyruvates are converted into ethanol or lactic acid. Thank you for listening to this recorded discussion and I hope that you learned something. Bye!